Welcome everyone, and thank you for joining our webinar today. So I'm going to start with a quick introduction about our company, Omics Playground, and then we'll introduce Vincent and Alexandra, who are going to be the main speakers today, and then let them take over. So, very quick introduction. We are Big Omics Analytics, and we develop platforms for the analysis of omics data that includes transcriptomics, proteomics, metabolomics, lipidomics, and so on. And Vincent has been using our platform for his analysis, so we are very pleased to have him talk about his results. So just a quick note for those who are not familiar with our platforms. Our goal is really to make bioinformatics analysis more accessible to people, to both democratize omics data analysis and also to reduce the amount of time that you need to obtain your insights. We think that our platform can really benefit most users as it can automate a lot of the routine analysis that people do with omics data. It's very interactive, but also user-friendly with more than 100 plots available. It also provides a unified omics data management hub. And crucially, it produces robust and reproducible results by combining best-in-class algorithms as well as more than 30 data sets for gene set enrichment analysis, as well as 6,000 published data sets that you can directly compare with your results. And also because it's a cloud-based platform, it's easily accessible across the world and allows you to visualize the data also with other colleagues anytime and any place you want. So now moving on, I just want to quickly introduce Vincent, I should say, and Alexandra. So they are currently both working at Uni Laval. Vincent is a professor and is specialized in especially respiratory disorders. He's very, very interested in hypoxia in particular, and how this can affect conditions such as apnea. And Alexandra, who was his PhD student back in the days, and is now postdoc also at University Laval, collaborated with him on this data set, in particular providing essential support for the analysis and interpretation of the data. So without further ado, I'm going to pass now the baton to Vincent, and please start sharing your presentation and introducing yourself and Alexandra more formally.
Okay, excellent. Thank you, Axel, for the introduction. And I would simply comment on that by saying for us, the use of the platform has been really instrumental in getting our data. I don't have any background or knowledge or will to learn coding, to go into R or Python or code, etc. So using this kind of platform for analysis of transcriptomic data is for me super. It's the way to go, the way to do, and it has been very, very helpful. So I will share my screen. This will be okay. And you tell me if everything is okay.
Yeah,
so
we can see your slides.
Okay, excellent. So without further delay, I will just want to maybe here this. So the title of this presentation is Omics Insight into Mito-Nuclear Imbalance in Intermittent Hypoxia. So I first want to, okay, who are we? We are respiratory physiologists with an interest in understanding physiological responses to chronic and intermittent hypoxia from the organism to the cells. And what do we do in relation to that? Well, we study models of adaptation and acclimatization to high altitude, as well as preclinical models of sleep apnea. And we used, of course, the big Omics platform to analyze specifically a set of single-cell RNA sequencing from the lung of mice exposed to intermittent hypoxia. So what is intermittent hypoxia? Intermittent hypoxia is a model of sleep apnea. And then you may ask, what is sleep apnea? Sleep apnea is a sleep and respiratory disorder, which is characterized by repeated episodes of complete cessation of breathing during sleep, up to 1 or 2 minutes, causing drops in arterial oxygen saturation and abrupt reoxygenation. It is considered as being clinically severe if there are more than 30 events for each hour of sleep. And the main risk factors include aging, obesity, and malnutrition. In terms of, this is a busy slide, but this is the clinical evaluation of sleep apnea, and you can look this pointer, yeah. No. Oh. Where can I find a here pointer? Okay. So this is a bunch of EEG recording from a patient sleeping, but the important part are here. This is the airflow, okay? And you can see that during what is called obstructive apnea, there is a complete cessation of airflow recorded here and here with chest and abdomen respiratory efforts, meaning that the patient is trying to breathe, but the airways are obstructed and the flow of air cannot go to the lungs. And in parallel, you have this red trace here, which is the arterial oxygen saturation. And you see that very clearly each apnea is associated to very profound and deep desaturation of oxygen, which goes on during throughout the night. And this is what the nights of this patient looked when looking for 6-7 hours of sleep. And you can see the arterial oxygen saturation with this very typical shape of very profound, repeated, and frequent desaturation. So this is intermittent hypoxia. Sleep apnea is one of the most frequent respiratory disorders. I said in the general population, it has been estimated that as much as 23% of women, 50% of men have moderate to severe sleep disorder breathing, which are characterized as more than 15 events per hour. And it is independently associated with hypertension, diabetes, metabolic syndrome, and depression, at least in this study. Why is it important? Because sleep apnea and intermittent hypoxia will affect many conditions. Patients with sleep apnea that are not recognized and treated are at risk for hypertension, stroke, heart failure, car accidents, neurological disorder, diabetes, metabolic syndrome, and it is also associated with mortality. All-cause mortality is increased, cardiovascular mortality is increased in sleep apnea patients, and cancer mortality is also increased. Considering lung disease, what are the links between sleep apnea and lung disease? It is estimated that 50 to 70% of patients with chronic obstructive pulmonary disease are apneic, which is called the overlap syndrome. So you have two clinical conditions that overlap, and each condition influence the developments of the other. Considering specifically COPD patients, it has been shown that sleep apnea increases exacerbation and death by up to 7-fold. Considering another very common respiratory disease, which is asthma, it has been demonstrated that 90% of patients with what is called difficult to treat asthma are also apneic. So there are some key references for this data. So on this background, intermittent hypoxia has been used for the past 40 years, 30 to 40 years, as a tool to mimic the intermittent hypoxia that are appearing in sleep apnea patients. And this is one of the first instruments that was used to model this condition, and in which you can see that there is a closed chamber and the rats can be put there, and simply the flow of air will be modified to expose intermittently the animal to bouts of hypoxia. You can see here the level of oxygen going from 21, which is normoxia, to 5%, I guess, here, repeatedly in number of episodes here during 15 minutes. And this is one event of hypoxia, and you can see clearly the association with arterial pressure, which goes up with each cycle, the heart rate also, which goes down and up, etc. And one of the first publication that I used this model, they've been able to show that there is, during long-term exposure, so several weeks of exposure every day to intermittent hypoxia for a few hours, and then you have a gradual increase of arterial blood pressure, which was very important at the time because hypertension is one of the most important comorbidity of sleep apnea. So showing that intermittent hypoxia in a rat model alone can increase arterial blood pressure was very, very important at the time. And the model has been used, it has been extremely successful to understand what happened in sleep apnea patients. And this is simply to show you that since the early 90s, we have gained a lot of understanding on the structures and the mechanism underlying increased sympathetic activity, increased blood pressure in models of intermittent hypoxia, and this involves elements of peripheral nervous system, such as the peripheral chemoreceptor, which detect oxygen levels in the arterial blood. And we also have clear, also incomplete data on the molecular biochemical mechanism underlying this activation. So what happened in the lungs then? Of course, the lungs are very responsive to hypoxia. There is hypoxia induces constriction of the capillaries in the lungs, leading to pulmonary hypertension. But any type of cells in the lung will see the hypoxia and respond to hypoxia, and you can see obviously in the alveolus these repeated drops of oxygen will necessarily induce cellular response. The animal model have been useful, and we're starting to see more and more studies exploring the effect of intermittent hypoxia on pulmonary function in rodents, mice, or rat models. It has been shown that it can induce expiratory flow limitation, which is a clear sign of non-function or poorly functional lungs. It increases collagen and enlarges air spaces. It reduces antioxidant defense by reducing the expression activity of key antioxidant enzymes, such as catalase, superoxide dismutase, which increases the level of reactive oxygen species, lead to lipid peroxidation, etc. It can activate the hypoxia inducible factor and NF-kappa, which are key transcription factor for hypoxic responses and inflammatory responses, triggering inflammation. And it has been shown to increase the risk of pulmonary hypertension, asthma, COPD, and fibrosis through lung damage. When we started the study on, we had the idea to make this single cell analysis in the lung. There has been one study which has been published so far, doing exactly the same analysis. So you can see this is a very typical protocol. The mice are exposed either to normoxia here for 9 days or to intermittent hypoxia for 9 days also. The intermittent hypoxia is applied specifically during the day when the animals are normally sleeping. And they are returned to normoxia during the night when they are active to mimic what happened in the clinics where patients are apneic only when they sleep. And then they took the lungs out and performed some extraction for RNA, single cell or bulk sequencing. But what was, and this is very classic in this kind of models, as you can see here, on day 10, the animals have been sacrificed and the lung samples without prior exposure to intermittent hypoxia on this day. So they have been left in normoxia during the night, in normoxia during the first part of the day, then sacrifice and sample extra. So you don't really have immediate cellular responses to intermittent hypoxia by itself. You have the effect of intermittent hypoxia and a recovery in normoxia. So you capture the cellular responses in a very specific state. This is fine. This is perfect, but it doesn't tell us what really happens at the cellular level during these episodes. So we designed our protocol to try to answer this question. So our protocol is 21 to 6% oxygen during the day, 30 events for each hour, 12 hours a day, 14 days, and all samples have been taken, I think it's 9 a.m. During the exposure to intermittent hypoxia. So there is 3 hours here of exposure when we took the samples. This is a very simple workflow. Should be noted that we pulled 3 mice by group. It was intended as preliminary data, preliminary experiments, and there was some financial consideration, so we decided to go with the easiest and less expensive design. So we pulled 3 mice here. We isolated the number of cells within each group, normoxic, hypoxic. Then we performed the sequencing, cell type identification. We retrieved 33 different cell types. And then we took this all number of cells and reconstructs what is called meta cells to work with the big omic platforms. So we have Infinal, which is a statistical representation of these cells. For those interested, go more in the details. We've so far published the paper as a preprint on the bioarchive server. The data are all available on the GEO website. And I will show you some pictures, some figures taken from this publication. But there is also, be aware that there are several whole series of supplemental tables, Excel tables that are presenting the data in a numerical way rather than figures. So we first perform. I will go very quickly on this aspect, quality control and cell identification using very, very standard tools. But all of this has been done outside big omic. And these are preliminary steps before this kind of analysis. But you can see here, for example, the markers used. So for every cell here, every line is a single gene. Yellow is the strong expression, and this is where you can identify different type of cells on your collection. So within Big Omics, we first started to make bulk analysis to have an idea of the different, what is called the DEX, different differentially expressed genes and gene set enrichments. And so this is directly a figure coming from Big Omics, representing the genes that are upregulated or downregulated. Red is upregulated, blue is downregulated in intermittent hypoxia compared to normoxia. So you can see here a bunch of genes that are upregulated. Some of them are related to mitochondria. And from this figure, you can also, from this data, you can also extract the more classical volcano plot, showing individual upregulated or downregulated gene by effect size, log2 fold change, and the Q value, which is equivalent to P value here, this statistical significance. So one of the first thing that caught our attention was the very strong concentration of mitochondrial genes here. This is a subunit of complex 1. This is a subunit of complex 5, another one for complex 1. This is a mitochondrial transporter, another one for complex 1, etc., etc. And when we looked here, downregulated, there were also some, these two MT ATP8 and D6, these are genes from the mitochondrial genome, which are downregulated. So we'll come back to this later. Then we performed here, this is the same two-dimension projection of the data sets, but instead of individual genes, these are gene sets. So within the platform, you have access to all collection of different gene sets from different data bank. And here, this is all of these presented here. And you can see again, among the most upregulated, oxidative phosphorylation, reactive oxygen species, peroxisome, but also myogenesis here, adipogenesis, fatty acid metabolism, and here the epithelial to mesenchymal transition. So these pathways are now represented here, which is also a classical view in this kind of analysis, classified by the net enrichment score. Next, we move to cell level analysis. So this is data here have been computed outside Big Omics, and you can see all the cells that have been identified, their name, of course, and the number, absolute and relative number, and in proportion to differences between normoxia and intermittent hypoxia. And interestingly, so a bit of warning here, we used pool data, so we had to perform some very specific analytical steps to be able to draw these figures. This was done outside Big Omics, but it gives you an idea if this number of cells are increased in intermittent hypoxia or decreased in intermittent hypoxia. What is really, really interesting, if you look here, so the relative number of cells that are increased in intermittent hypoxia, these are all cells that belong to the alveolar units. You have here alveolar macrophages, 81 alveolar type 1, fibroblast type 2, capillary type 1, etc., which are all increased in intermittent hypoxia. And this is really interesting because we know from previous studies that in mice exposed to hypoxia, whether it's continuous or intermittent, the lungs are growing. They grow bigger lungs as an adaptive response to hypoxia. So here we have an increased number, and we can see that there are specific cell types in the alveolar units, which are more numerous in intermittent hypoxia. Here on the bottom side, you have the cells that are their numbers are downregulated in intermittent hypoxia, and these are mainly immune cells and lymphatic and cells from the lymphatic system. I will go also later on this. So next step, from big omics, you can have an idea of the number of genes which are upregulated or downregulated for each cell type. So we retrieve the table, and simply this is a graph pad presentation of this data. But what was super interesting to see from this data is that you have roughly three different groups of behavior in response to intermittent hypoxia. You have these cells here, airway smooth muscle cells, lymphatic endothelial cells, and arterial endothelial cells, in which they have a huge number of genes that are down or upregulated. Then you have a whole bunch of cells which have an intermediate number of genes upregulated or downregulated, and some cells which are very low responders to intermittent hypoxia. So we took these three types of cells, airway smooth muscle cells, which are so important in the regulation of airway resistance, obviously key contributors in asthma, for example. The lymphatic endothelial cells, which are also super important in regulation of immune function in the lungs, and the arterial endothelial cells, which are also very important for hemodynamic control of lung perfusion. So again, upregulated, downregulated genes, but you see here these mitochondrial genes, which are also present here, and DUF, ATP, etc., COX, present here. This is just an example of a low responder. And so from the Big Omics platform, we retrieve these figures. Again, this is the same presentation that I showed you in the first series of data with the bulk analysis. This is the UMAP clustering of gene enrichments in airway smooth muscle cells, lymphoid and arterial endothelial cells, and you can see that visually from here you can already spot some similarities. This region is enriched in all three types of cells, for example, and this is where we have the mitochondrial gene sets, reactive oxygen species, etc. And then we make this figure, showing the enriched pathway in these different cells by size of the size is the number of genes which are involved in this pathway that are upregulated, and here the score, the strength of the enrichment. So you can see, for example, in airway smooth muscle cells, they respond to intermittent hypoxia by increasing the expression of genes involved in myogenesis. So this will be very highly relevant for disease like asthma, for example. Epithelial to mesenchymal transition is also increased in these cells. And for example, here you have oxidative phosphorylation, which is increased in lymphatic and arterial endothelial cells, but not in the airway smooth muscle cells. So you get insight into how the cells individually respond to intermittent hypoxia, which is really, really interesting. So we've gained, as I said, what we've gained here from this analysis is that we have knowledge about the absolute and relative number of cells that are altered by intermittent hypoxia, the absolute number of genes that are differentially expressed by intermittent hypoxia, and the cell phenotypes linked to lung function and respiratory disease. Then the next, something that is also very interesting is to go to the pathway level analysis. So instead of looking at individual genes, you really focus on the pathways that are up or down regulated. And what was absolutely stunning is the number of cells. So here, each line is a different cell type. Each column, sorry, is a different cell type, and each line is a different pathway. And you can see that the number of cells, there is a really huge number of cells in which oxidative phosphorylation is upregulated. So we look more closely. So this is something that we retrieved from big omics. We looked more closely at the pathway, and so you can see here complex 1, complex 2, complex 3, complex 4, 5 UCPs and translocators of the oxidative electron transport chains, and colored by fold change of the gene in response to intermittent hypoxia. So you can very easily see that complex 1, 2, 3, and 4 see upregulation of different subunit components in response to intermittent hypoxia. I mentioned briefly in the first slide that, for example, when you look at the volcanoplots of the differentially expressed genes in intermittent hypoxia with the bulk analysis of all cells combined together, as I said, we have these genes which are mitochondrial genes contributing to the electron transport chains, but these are genes coded by the nuclear genome. Okay, and you have also here downregulated these genes, which are also contributing to mitochondrial complexes, but this one are coded by the mitochondrial genome. So there is kind of this balance or imbalance between nuclear and mitochondrial gene expression. So what is the mitochondrial DNA? Well, it's a very simple loop with a very limited number of bases. And in total, I think 13 genes that are coded by this genome that are part of the mitochondrial complexes or ribosomal RNA. And I didn't really knew what to do with this information. So I tried to go in the literature a little bit. And one way to represent this data is to use the log2 fold change. So in all the cells that show Mito-Nuclear gene downregulation and nuclear mitochondrial gene upregulation, we took the mean log2 fold change in lymphatic endothelial cell, arterial endothelial cells, and again here you recognize the cells from the alveolar units, the fibroblast, the alveolar type 1, etc., capillaries, and you can represent this by simply showing the up or down regulated fold change. Then simply this index value, which is the sum of these two bars, will give you the strength of the mito-nuclear imbalance. So on top of this, you have the lymphoid endothelial cells. I've not commented on this, but they are also super important in lung diseases. The lymph system in the lungs drain fluids and immune cells. And there have been studies showing, for example, that in fatal asthma cases, you have a decreased number of lymphoid cells and lymphoid vessels, which it's easy to understand in case of a severe asthma attack. Then you have infiltration. You can have infiltration of immune cells, and the system needs to drain these cells and fluids outside of the lungs. And if this capacity is reduced, then it will aggravate furthermore the crisis. So it makes perfectly, in terms of disease progression and severity in intermittent hypoxia, it is really concerning that the lymphoid endothelial cells are responding so strongly to intermittent hypoxia. So to go back to the mitonuclear imbalance, This is defined as an imbalance between the expression of nuclear and mitochondrially encoded mitochondrial proteins, I told you. And what is really interesting is that it activates this imbalance of gene, activates the mitochondrial unfolded protein response. And surprisingly, interestingly, whatever you want, it has been shown to extend the lifespan in C. elegans. And we also have emerging data that link these responses with lung function in ALS and disease. So what we've gained from this analysis so far, the key points, if you want to take away, that the most prominent responses, so we have identified the cells that have the most prominent responses to intermittent hypoxia in terms of differentially expressed genes. These are the airway smooth muscle cells, the lymphatic endothelial cells, and the arterial endothelial cells. We have been able to identify strong enrichments, particularly relevant for pulmonary disease progression. For example, myogenesis and epithelial to mesenchymal transition in airway smooth muscle cells, oxidative phosphorylation and reactive oxygen species in arterial endothelial cells. We have identified the most common enriched pathways across all the cells that we have that are present in the lungs. And the most notorious one is oxidative phosphorylation, expressed in 20 different cell types. DNA repair, I'm not commented on that, but it's upregulated in 8 different cell types. MYC target V1, also not commented. This is a non-oncogene involved in cellular multiplication, division, etc. Upregulated in 7. And the ROS pathway and the epithelial to mesenchymal transition, which is upregulated in 4 different cell types. We have also shown that there is an increased number of alveolar cells in response to intermittent hypoxia, probably linked to adaptive responses to hypoxia in mice. And we have identified the Mito-Nuclear imbalance that is present mostly in alveolar, in arterial and lymphatic endothelial cells, and also cells of the alveolar capillary units, the fibroblasts, the alveolar type 1, capillary, etc. And that's, so of course, we still have a lot of work to continue to understand these responses in the future. I will be very interested to have one set of data, for example, trying to capture what happens as we did here in immediate response to intermittent hypoxia, but also the progression during recovery. I think this is very important in diseases such as intermittent hypoxia. So we still have a lot to do to try to understand the whole picture. So thank you very much for your attention. And we can take
Vincent,
some questions.
Absolutely. So thank you so much for an excellent presentation. I already have a couple of questions, but of course, I would like first to hear from our audience if they have any questions. So what you can do, you can raise your hand. I don't know if you know how to raise your hand on Zoom, and then we can call you out, and you can activate your microphone and ask your question directly to Vincent. Any questions? Well, maybe while I give people time to think, if they want to think about more questions, I had a couple that arose from one that arose from your conclusions, and that I saw the upregulation of MYC genes and other oncogenes associated with apnea, or at least hypoxia. Do you think this could actually translate in an increased risk of developing lung tumors?
Probably. Well, we've not looked very deeply into this pathway because I don't know it. So, but there are clearly links between sleep apnea and numerous cancers. But the data from the literature on this, the clinical data are not so rather scarce, but there have been some few studies showing links. I've seen a recent paper showing that it increases sleep apnea, increases cancer mortality. So there is certainly some elements here that can help understand these things. Sure.
Excellent. And the other question I had was looking at the profiles of up and down regulation. Have you thought about possible interventions, obviously beyond anti-inflammatory drugs, that could benefit patients?
Huh. This is another really interesting question. Yeah, I mean, in the end, but the utility of this data, the ultimate utility of this data could be to help better understand which are the pathways that could be targeted um, to avoid deleterious consequences in patients that, for example, in overlap syndrome, when you have a patient with COPD and sleep apnea, then maybe there would be specific drugs that could be designed to help avoid that the sleep apnea of the patients aggravates the respiratory condition, for example. But I've not looked, for example, I guess in the back of your mind, you have some idea that the data can be used to try to identify therapeutic molecules that target specific genes or expression or pathway, etc. That will be very interesting. Yeah. To try
Yeah,
to
it's
yeah.
it's interesting if maybe there are already validated drugs, already FDA approved drugs that could be repurposed.
Yeah, yeah, that's a very
So,
interesting suggestion. Yeah,
uh, uh, uh. Well, it's something you can do with the platform. So,
yeah. I know. I've looked. I've seen this. But yeah.
yeah. I'm not saying that you should immediately go and say, yeah, let's use this. Obviously it requires
Mhm,
testing and you never know what the side
mhm,
effects
mhm.
are, but
Yeah.
obviously it's all very interesting information. And another question that I had based on the beginning of your presentation is when you were talking about the association between um, neurological disorders or cognitive decline and the development of apnea. I wonder, could there also be a reversed influence, that is, changes in hormone levels or impairment of neuronal signalings? Could it induce the development of apnea in patients?
Yeah. Sure. And in disease, for example, neurodegenerative disease, Alzheimer, Parkinson, etc., but also other kind of neurodegenerative diseases. So you can have the neuronal network that control breathing can be modified or affected, altered by the conditions, so that it become less effective and can induce apnea, etc. Yeah,
Yeah, it's quite interesting
yeah,
how everything can be interconnected. So
yeah, yeah.
I just wonder, mm, yeah, as there are there any studies that associate possible defects in the neural systems with apnea that you know of, or is it still pure speculation?
No, there are certainly, and the most common case is heart failure. So this is a heart disease, but it completely changes. It's a very strong influence on the peripheral chemoreceptor that I mentioned in the introduction, and drives respiratory instabilities and induces central sleep apnea. So this is the same kind of apneas, but without the obstruction. So this is really the central respiratory neurons which stop working and stop sending their signals. So respiration stop during, but it has exactly the same effect on intermittent hypoxia. So this is one of the prominent example of sleep apnea induced by a specific disease condition not related to obstruction.
Okay, so well, the reason questioned it's more about the use of the platform, but maybe it would be great if you can address it, Joseph, Vincent, play based on your experience, and this comes from Bilal, and is asking how transparent to the user and how flexible is the platform regarding computation, the computation it does to produce the visualization that you showed.
So you can you can choose the different tests for example, definition of DIGs, differentially expressed genes. So you have different tests that you can choose. I guess for when you import the data, and then when you visualize the data, so you can select these different tests, and you provide some recommendation. But then it's free to the user to test different approaches. Print out to text windows as steps and run to modify algorithm setting with a graphical user interface.
Yeah, that's
That's
that's how you
advanced level.
interact.
Yeah, a bit technical maybe.
You cannot
No, this is
like, if I can just say something.
yeah, go on.
You cannot directly modify the algorithm. However, you can just change some specifics that are available in the platform. And everything that is done it's kind of described, so you know which there is a tutorial, or there is also a written sheet where everything that is used, which script, which version, etc is described. So you can read this and you have all the more specific information, but you cannot directly work onto the script.
Yeah, and for each step, each figure, etc., you have access to the data, the underlying data, so you can download CSV or Excel files, and we've done this a lot. So for each this is why I saw the number of supplementary tables that are in the paper, because for each figure there is one or several Excel files that really have all the data associated to the figure.
Right. So
Does that answer your question? Or yeah, please go on.
yeah. So you can really understand what you're doing
Yeah.
and why you're making the choices that you make, especially when your work comes up to scrutiny. So yeah, it was in that sense. Thank you very much.
Yeah, no, as Vincent and Alexandra already mentioned, there is an extensive, not just tutorial, video tutorial, but also an extensive description of all the methods, the parameters that we use, that basically tell you exactly, like if you choose trend lemma, how is the trend lemma working in identifying differentially expressed genes.
Yeah. And Axel is always one email away from a call or specific questions, and have been very reactive and supportive in when needed. So that was also very useful.
So there is a question from Blaine, and that's definitely more for you, Vincent. Have you ever looked at tissues other than lungs to see if these transcriptional effects are a more general response?
Sure. So far, we looked at liver, and but this is slightly different protocol because we also add obesity, intermittent hypoxia, sex, male, female, etc., but not in single cell sequencing. So far, in terms of single cell sequencing, we are really focused on the lungs. But of course, yeah, it will be really interesting to see if these responses are shared between different tissue or they are so tissue specific. I guess if I have to guess something, I would say that we will see some pathways shared between tissues, and we will see which are different. But this is just a wild guess. So.
And there is one more question for both you and Alexandra, and that's from, I hope I'm not butchering the name, Jaime Pablo. And the question is, do you happen to know if the same pathways are shared between the animal model and humans? Also, do you think targeting the mitochondrial function could improve the outcome of these patients?
No to the first question because there's no tissue bank or tissue analysis from sleep apnea patients, for example, in lungs samples, etc. But of course, we can we can try to. I mean, given the role, for example, this very strong response in airway smooth muscle cells and the link in the literature between sleep apnea and asthma, for example, this can be, we can imagine that the airway smooth muscle cells in human will respond somehow to intermittent hypoxia, maybe not exactly similarly, but something close. And targeting the mitochondrial function to improve the maybe. But the field of the field of mito-nuclear imbalance is relatively young. I mean, when I search in the literature about the first time this concept has been coined was about 10 to 15 years ago. So this is, and there are a few labs really trying to understand this at the core. So it certainly will evolve further in the future. Yeah, and that's something to pay attention to for sure.
If you have anything to add, otherwise, I think.
May I have one question?
Yeah, go on. Yeah, Jorge.
Yeah, thank you. Sorry, it took some time to find where I have to raise the hand. But well, basically, congratulations for the science behind. Well, I know very well to Vincent, and I know what he's doing. So it's not surprising the nice results he got. But I'm here more for the technique rather than for the results you obtained, because what you're doing cell sorting to do all this analysis, right? And what we took in our experiments were pieces of brain to do proteomics. And in the proteomics platform we have here at Laval University, and we have a list of molecules that are up or down regulated. And with these molecules, we did the classical analysis of volcano plot, heat map, protein-protein interaction, the gene ontology, biological process also. But somehow we were stuck there. And I would like to know how still I can do with this data using this omics platform. How I still can take more profit from this data. And that is my question. Thank you.
Well, maybe I can answer that one. So it's a very good question, and the real power of our platform comes from the fact that you can put your data set in a larger context. And we can do this in three ways. First of all, we have a large collection of public databases of gene sets and pathways. Secondly, we have integrated into our platform drug-related collections such as the L1000 drug connectivity map, the drug sensitivity map, perturbation map. And thirdly, we have more than 6,000 experiments from the public domains that also include a lot of lung diseases, for example, that you can use to see the context of your data across these public experiments. Now, if you're interested, would be more than happy. Maybe we can have a quick demo. I think I do have some relevant data sets that I could show you. So maybe you can get in touch with us and
Yeah,
we can talk more about that.
yeah, great. Thank you. Thank you very much. I will do so. Thank you.
Oh, you're
Thank you,
welcome.
Vincent. Thank you, Alexandra.
Thank you.
Thank you.
And I think we are close to the end, so I'll just put a slide up just to remember people or users and people who are not users who are interested of knowing more about the platform that we are going to have a Q&A session on the 11th of December. We'll put out more details about the exact time, and if you wish to contact us, you see here our website and our contact address. So I wish to thank you, everyone who joined, and I wish particularly to thank both Vincent and Alexandra for their great presentation, and hope to see you again at our next webinar. Thank you.